So it is my pleasure to introduce Dr. Stacey Finley, who is the inaugural winner of the Leah Edelstein-Keshet Award. This new award was named after the SMB's first female president and is given every 2 years. This year's award recognizes a woman in an early stage of her career who is making exceptional scientific contributions to mathematical biology, developing a strong independent research program, and exhibiting a continuously high level of scientific leadership. So congratulations. So I want to tell you a little bit more about Dr. Finley. Dr. Finley received her bachelor's degree in chemical engineering from Florida A&M University in 2004, and her PhD in chemical and biological engineering from Northwestern University in 2009. From 2009 to 2013, Dr. Finley was a postdoctoral research fellow at Johns Hopkins University School of Medicine. Dr. Finley currently holds the Gordon S. Marshall Early Career Chair in the Viterbi School of Engineering at the University of Southern California. Dr. Finley has been awarded many prizes and fellowships in her short career for both her outstanding research and her role in mentoring young individuals. And some highlights are, of course, the Leah Edelstein-Keshet Prize, but also the NSF Graduate Research Fellowship and the NSF Career Award. Dr. Finley's research applies a systems biology approach to develop computational models of angiogenesis, metabolism, and immunotherapy in the context of cancer. And in today's talk, Dr. Finley will speak about angiogenesis in tumor breast tissue. Please join me in welcoming Dr. Finley.
So thank you very much for the introduction, and thanks to all of you for coming to the lecture. It's really a great honor to be the inaugural recipient of this prize in honor of and recognition of Dr. Leah Edelstein-Keshet. It's just a privilege to be able to tell you about some of the work that my lab is doing at USC, and I'm really excited to have this opportunity. So what I'd like to talk to you about today is the work that we've been doing in developing systems biology models that allow us to gain more quantitative and mechanistic insight into the angiogenic state of breast tumor tissue. And so before I begin in talking about the research, I really wanted to give you an overview of my lab at USC, where one of the primary research goals is just to be able to predict the dynamics of various biochemical networks, and particularly predicting these dynamics in the context of pathological conditions, and right now we're focusing in on cancer. And so if we have this quantitative and predictive tool that we can use, we can better understand the effects of existing molecularly targeted therapies, as well as provide insight into what should be novel therapeutic strategies. And so we can use a lot of different systems biology tools to do this. So we use chemical kinetic modeling, really drawing off my training and expertise in chemical engineering, as well as putting together the model in an automated fashion. And I'll talk a little bit more about this later. And then also doing model optimization so that we can parameterize the model and fit the model to experimental data. And then lastly, my lab has begun to do quantitative experimental studies. This is really important and exciting for me so that we can have the data that's needed to build the model up, as well as to go back and test some of the model predictions. And so we're doing this work in a number of different applications, again, all with the same types of tools, but studying different aspects of cancer biology. And in one case, we're looking at T-cell signaling, where T-cells have been engineered with chimeric antigen receptors, and this is in the context of cancer immunotherapy. We have a recent paper that describes a kinetic model of the dynamics of pancreatic cancer metabolism. And then again, what I'll talk to you about today has to do with our model in angiogenesis signaling, specifically in breast tumor tissue. And so I want to separate my talk into two parts, where I'll tell you first about how we're predicting the angiogenic balance in breast tumor tissue, and this is work that was recently published at the end of last year. And then lastly, I'll present a more preclinical model of tumor-bearing mice and some of the insights that we can try to gain in terms of predictive biomarkers that can predict the effects of anti-angiogenic therapy. And this work is under review, so it's in submission right now. If any of you are reviewing this, I would love to talk to you more about it. All right, so we'll start with just sort of an overview of what angiogenesis is. So this is the formation of new blood vessels from pre-existing vasculature. This is a really important process that happens in development, for example, in organ growth. However, as adults, most of us don't have a lot of new blood vessels that are being formed on a regular basis. But there are very normal physiological processes that require the formation of new blood vessels, for example, in reproduction, in wound healing, and also in exercise. And so the extent of this vascular network and the size of the network is really governed by a balance of promoters and inhibitors of angiogenesis. And a disruption in that balance can lead to a number of different pathological conditions. And some of those conditions are characterized by having too much new blood vessel formation, and that's what happens, for example, in the case of cancer and also in retinopathies, whereas other diseases are characterized by having insufficient angiogenesis, and that's what happens, for example, in the case of peripheral artery disease and stroke. And so I put in ellipses here because there are more than 70 different diseases that are related to angiogenesis and could possibly benefit from angiogenesis-based therapies. So having a quantitative framework that we can use to better understand the dynamics of these signaling pathways and processes could be very helpful in a number of different contexts. So my work is focused in on cancer. Again, this is a case where too many blood vessels are being formed. And so one of the very important promoters and regulators of angiogenesis is called vascular endothelial growth factor, or VEGF. And this is a strong promoter of angiogenesis that really acts in response to a number of different stimuli, biochemical stimuli, mechanical stimuli, and then also in response to hypoxia, which is the condition of having low levels of oxygen. And in response to all of these stimuli, VEGF can promote cell proliferation and migration, and also cell survival. And this figure sort of illustrates what the role of VEGF is. So you can consider this purple blob to be a hypoxic tumor cell, and it recognizes that it's in low levels of oxygen through hypoxia-inducible factor 1, or HIF1. This is a transcription factor, a molecular oxygen sensor for the cell, which is actually a transcription factor for a whole host of genes that it's able to activate, including the VEGF gene. So that leads to the transcription and translation of VEGF, and then the protein, which are these little blue dots, are being secreted by this hypoxic tumor cell. So the protein VEGF can bind to its receptors that are on the surface of existing endothelial cells. That leads to activation of the endothelial cell so that it's able to break through and break down the surrounding basement membrane and move through the extracellular matrix, and then proliferate and move up the VEGF gradient towards higher concentrations of VEGF. And so for this important role of angiogenesis, and specifically VEGF in this process, many cancer therapies are aimed at inhibiting this VEGF-mediated signaling. And those are called anti-angiogenic therapies. Okay, so as a postdoctoral research fellow in Dr. Sasha Popo's lab at Johns Hopkins, I really had a great opportunity to use mathematical modeling to gain more insight into the strategies, the anti-angiogenic strategies that are aimed at inhibiting VEGF-mediated signaling. And some of this work, we were able to better understand the effects of drugs, properties, and also tumor-specific properties in influencing the response to treatment. We were able to very nicely compare the data to some clinical measurements as well, and also, for example, understand some of the drug pharmacokinetic properties. But since starting my lab at USC, we've been expanding on this work to not just look at the promoter of angiogenesis, but other angiogenic factors, and specifically looking at an inhibitor of angiogenesis called thrombospondin 1. And in this work, we can now be able to predict the balance in tumor tissue, and as I'll talk to you about today, also have this preclinical model where the volume of the tissue specifically and explicitly depends on the angiogenic signal. So I mentioned that angiogenesis really is this balancing act. It's the balance of promoters and inhibitors that determines and influences the extent of vascularization, but it's also this balance that plays a really important role in any therapeutic strategy that's aimed at inhibiting angiogenesis. So we want to take care to focus in on this balance, and we've been looking at anti-angiogenic factors, endogenous factors, and one of those is called thrombospondin 1. So I want to just walk through the many different ways in which thrombospondin 1, or TSP1, is able to inhibit angiogenesis. So I'm going to apologize up front that there are a lot of acronyms that I'm going to introduce to you, but really the takeaway message is to understand and use these models to gain more insight into the biology of the system and how we can better understand the heterogeneous response to these anti-VEGF and anti-angiogenic therapies. Okay? So I mentioned that VEGF can bind to its receptors, and then it promotes proliferation and migration signaling through nitric oxide and also through phosphorylated ERK. Well, thrombospondin 1 can bind to its own receptors, namely CD47 and beta-1 integrins, in order to antagonize VEGF-mediated signaling. So this is one mechanism by which TSP1 can inhibit angiogenesis. Alternatively, TSP1 can bind to another receptor called CD36 in order to promote caspase 3 activation, which mediates apoptosis or programmed cell death. So this is another way that thrombospondin 1 can oppose angiogenesis. And very interestingly, this molecule, thrombospondin 1, can bind directly to VEGF to form this complex that can then be degraded in the extracellular space by the action of proteases, or that protein can bind to another receptor called LRP1 and form this non-signaling complex that the cell can then internalize. And so this is a way to remove VEGF from the extracellular space. And the VEGF receptors can couple to thrombospondin 1 receptors, to which the ligands can come in and bind and form many different permutations of another set of non-signaling complexes, which can then again be internalized by the cell. And then lastly, some of the VEGF isoforms, some of the forms of VEGF can bind to glycosaminoglycan chains in the extracellular matrix, in the ECM, and thrombospondin 1 can inhibit the activity of the enzymes, some of the enzymes that are working to release VEGF from the matrix. So again, altering the availability of VEGF in allowing it to bind to its receptors. So when we see this sort of very intricate and complex network, we really want to understand, well, what are the dynamics, what are the kinetics of these different processes of binding and unbinding and complex formation. And so what we want to basically do is have this mechanistic model of VEGF and thrombospondin 1 in tumor tissue, first of all so that we can understand, well, what is the distribution of these angiogenic factors, but also so that we can use the model as a platform to understand and investigate different strategies that mimic the thrombospondin 1 anti-angiogenic mechanisms. And these are called TSP1 mimetics. So that's what I'll talk to you about first. So we have this model which represents a breast tumor that's 4 centimeters in diameter. It's a compartment model, which means that we don't consider spatial variations, but we do consider localization, right? So we have endothelial cells separated from the tumor cells by a collagen that's representing the extracellular matrix. The endothelial cells and tumor cells are expressing VEGF receptors as well as thrombospondin 1 receptors, and also they're the source of various soluble factors, including VEGF and thrombospondin 1, and those enzymes that I mentioned release VEGF from the matrix. So this is a highly simplified schematic just for you to see some of the molecular players. What actually we do is have 130 ordinary differential equations that describe how the concentrations of all of these molecular species evolve over time. And so we're not actually writing out all of these ODEs by hand, but what we take advantage of is this software called BioNetGen, which is a rule-based modeling approach that can automatically generate the reaction network. And what we do is say, okay, we know that VEGF can bind to one of its receptors, and perhaps it doesn't matter if that receptor is coupled to a thrombospondin 1 receptor, so we can put in these specific rules, and then the modeling framework will generate all of the differential equations. Okay, so we know, of course, with any model that we have to have kinetic parameters, which we take from the literature. VEGF signaling has been studied for many, many years, and we take advantage of that broad literature base. And then also we have to specify the initial conditions, and so that's primarily the receptor density, so how many of these receptors are present on endothelial cells and tumor cells. So we can extract some of that information from the literature, and we've also been able to quantify on a cell-by-cell basis how many receptors tumor cells and endothelial cells are expressing in my own lab at USC. But of course we have to make some assumptions in terms of what these kinetic parameters are, and so what we do is to perform a sensitivity analysis which helps us to quantify what are the influential parameters and how do they modulate the model predictions, specifically the concentrations of VEGF and their complexes. And so what we do then is to say, okay, for those influential parameters, We're going to specify some range over which they can vary, and then sample from that range according to a Monte Carlo analysis, basically. Sampling from that range and then running the model 5,000 times with all different combinations of these possible parameter values so that we can understand what is the robustness of the model predictions. And so we're not saying, well, there's just one correct answer, but actually a range of predictions that the model can produce. So when I show the results, I'll show the mean value of these 5,000 simulations as well as the standard deviation. So you get a feel for the robustness of the model. Okay, so some data that's not readily available in the literature are the rates at which those soluble species are secreted by tumor cells and by endothelial cells, or what is available is varying pretty widely. And so we tune these specific rates of secretion for VEGF, thrombospondin 1, and also the matrix metalloproteases, those enzymes that release VEGF from the matrix. And so we take a lot of data, and I'll admit that not all of this information is taken from breast tumor tissue, but there's really just a paucity of data, and so we try to get our hands on as much information as we can. And so we take these measurements from different types of cancers and tune the secretion rate such that the predicted concentrations of these soluble species in the interstitial space between the tumor cells and the endothelial cells matches the experimental data. And so we have to do that tuning in order to make sure that we have a predictive model. And so we can do a pretty good job. Again, this is the mean value of our 5,000 simulations, and this is the standard deviation as well. So now we can go forward and say, okay, let's try to gain some novel quantitative information. It's possible to measure the interstitial volume, or excuse me, the interstitial concentrations of these species, but what's more difficult to measure is where are these species bound? What are they localizing to? And so we know that these VEGF and thrombospondin 1 can be unbound or free. They can be bound to their receptors, bound to one another, and so on. And so what we can use the model to understand is what is the distribution of thrombospondin 1 and VEGF throughout the tumor tissue. And so two things that I want to draw your attention to are that most of thrombospondin 1 is in this cleaved form due to the action of a protease called plasmin. So plasmin can readily cleave thrombospondin 1 and basically convert it to an inactive form so that it's not able to oppose angiogenesis. It can't bind to VEGF. It can't bind to its receptors. So more than 60% of thrombospondin 1 is in this inactive form. Again, the bars are the mean value, and then the errors indicate the standard deviation of those 5,000 simulations. Okay, and that's in stark contrast to what VEGF is doing. So more than 80% of VEGF is bound to its receptors, able to actively promote angiogenesis and intracellular signaling required for proliferation and so on. And so this really tells us a lot about the angiogenic state of the tumor tissue, why it's poised into a pro-angiogenic state, namely because most of VEGF is bound to its receptors, most of thrombospondin 1, that inhibitor, is in an inactive form. Okay? So we can look in a little bit more closely at this receptor-bound form of both thrombospondin 1 and VEGF. So less than 10% of thrombospondin 1 is bound to its receptors. And we can have maybe a more concise way to be able to quantify this angiogenic state. And so we've defined what's called the angiogenic ratio, which we define as the ratio of the pro-angiogenic complexes involving VEGF compared to the anti-angiogenic complexes involving thrombospondin 1. And at steady state, this ratio is 8.7, which means that there are nearly 9 times more pro-angiogenic complexes compared to anti-angiogenic complexes. Again, giving us more quantitative and mechanistic insight into the angiogenic state of the tumor tissue. Okay? So we can use this model as a way to not only predict the concentrations, but also the distributions of these angiogenic factors, which are difficult to measure experimentally. But we want to move a step forward and say, well, can we now use the platform as a way to test different anti-angiogenic strategies? Specifically those strategies that mimic different aspects of thrombospondin 1's anti-angiogenic mechanisms. And so what I'll show you now are simulations where we look at and estimate what is the percentage of thrombospondin 1 that is bound to its receptors. And this is after administering a mimetic, a TSP1 mimetic, that mimics different aspects of thrombospondin 1. So in this case, I'm showing the results for the CD47 binding mimetic, We first allow the model to come to steady state, and then at day 1, we simulate an injection directly into the tumor tissue, and then another injection 3.5 days later, so 2 injections per week. And then we see how does this percentage of the receptor-bound thrombospondin 1 evolve over time. Again, the solid line is the mean value from those 5,000 simulations. The shading indicates the standard deviation. Again, so you can have a way to appreciate the robustness of the model predictions. Okay, so we can see that once you add this drug, what we're including now is both endogenous thrombospondin 1 and the exogenous drug agent that we've just now injected at day 1 and day 4.5. And we can see that this drug has a strong impact on increasing the percentage of receptor-bound thrombospondin 1, which we should expect. But we also can look at that angiogenic ratio and see how that varies in response to administration of this drug. And so we can look at the ratio between the pro-angiogenic complexes compared to the anti-angiogenic complexes. At steady state before any drug is given, it's 8.7, which I previously mentioned. But then as soon as you administer the anti-angiogenic complex, which is the thrombospondin 1 mimetic, then the ratio significantly decreases. And actually it becomes less than 1, which means that now the number of anti-angiogenic complexes exceeds the number of pro-angiogenic complexes. So this CD47 binding mimetic is very much able to strongly shift the balance in opposition of angiogenesis. And we can do the same for other thrombospondin 1 mimetics. I do want to just bring your attention to this last set of figures, which is for the CD36 binding mimetic, and just to show that it's not nearly as effective in increasing the percentage of receptor-bound thrombospondin 1. And although it does decrease the angiogenic ratio, it's not as strongly decreased as the CD47 binding mimetic. And this is interesting because it's actually the CD36 binding mimetic, this last set of panels, that has been most heavily pursued in the clinic, leading to phase 1 and phase 2 clinical trials. But it has not been shown to be very effective, and perhaps the model is helping us understand some mechanistic information as to perhaps why we see those disappointing results in the clinical trials. So the model allows us to have this predictive framework to test the effects of different anti-angiogenic treatment strategies, specifically those in this case that focus on mimicking the action of thrombospondin 1. So even what I just mentioned just now, this vignette about CD36 binding mimetic, shows that just targeting a single factor or a single pathway has just limited success in terms of reducing tumor burden and also having a long-term effect on positive outcome for the patient. And so instead of targeting single pathways or single entities, now it's more exciting and more probable that we should focus on combining strategies. So in this case, perhaps we can combine anti-VEGF strategy with the thrombospondin 1 mimetic, and that's actually what researchers are doing now. There's a recent clinical trial that looks at this dual inhibition treatment, so 2 ways of inhibiting angiogenesis, and they did see some effect in a subset of patients, but not across the entire population. So again, we can try to use our model to better understand this heterogeneous response in terms of the dual inhibition and anti-angiogenic treatment strategy. Okay, so what we did is now we don't just have a tumor tissue, but we expanded the model to also include normal tissue as well as blood, so that we can look at trafficking between the compartments and the distribution of the soluble factors and the drug throughout the body. So now we're mimicking the entire breast cancer patient. And so what we want to do is basically recapitulate or simulate that same clinical trial. And what they did is to have the anti-VEGF agent called bevacizumab, which they administer intravenously, so directly into the blood compartment, once every 2 weeks. And then the ABT510, which is a thrombospondin 1 mimetic that binds specifically to CD36, and they administer this drug twice daily. And this higher frequency is required because the ABT510 is just a short peptide sequence that's actually cleared very quickly from the blood. And so there's a higher frequency needed in order to maintain a particular desired level of the drug in the blood. Okay, so we can use the model to perform different simulations, and it's just essentially what we want to understand is what is the role of tumor heterogeneity in terms of how it influences the response to treatment. And so specifically, we can alter the number of VEGF and thrombospondin 1 receptors in the tumor compartment itself, We know that there are a lot of different aspects to tumor heterogeneity, and so here we're just focusing in on the angiogenic receptor expression profile. And so we can vary the number of receptors on tumor cells and on the diseased endothelial cells within the tumor. And then again, vary these receptors, perform Monte Carlo sampling so that we can have an idea about how changing the receptor number influences the response to treatment. And so we can predict not only the thrombospondin 1 and VEGF concentrations, but also that angiogenic ratio that I mentioned previously. Okay, so these are the results from solving those nonlinear differential equations after we administer the anti-VEGF agent combined with the thrombospondin 1 mimetic. So we first again allow the model to come to steady state, and then at day 1 we simulate that injection of both the bevacizumab and the mimetic, just mimicking again the protocol outlined in the clinical trial. And so we can look as a function of time how the concentration of VEGF and thrombospondin 1 changes with time, and also the angiogenic ratio. So VEGF as well as the angiogenic ratio are very sensitive to this treatment protocol, so you can see that they change pretty drastically, whereas thrombospondin 1 is pretty robust to change. And this is what's happening in the tumor tissue, but also we can see and solve the equations to see what happens in the blood compartment and also in the normal compartment, which really is similar dynamics compared to the tumor tissue as well. So we can look at these profiles over time. We also wanted to have a more concise way to characterize the response. And so what we can look at then is what we're defining just as the fold change of these quantities, the concentrations as well as the angiogenic ratio. And we're basically taking the time point at the end of the drug, at the end of one cycle of the treatment protocol compared to the pre-treatment level as the fold change in the concentrations and in the angiogenic ratio. And then specifically for the angiogenic ratio, we also characterize the response to treatment using the area under the curve, mainly because it depends on what point we decide as being the post-treatment level. And so here we want to understand the integration over time of this effect, and so we look at the area under the curve in that case. So even from here, you can perhaps appreciate that there are some differences in the response level, so that's indicated by the mean value, but also the standard deviation. And so that heterogeneous response is illustrated even more clearly, I think, in this box and whiskers plot. So we can look at how the fold change is, again, the post-treatment level compared to the pre-treatment level after one cycle of treatment, and how that changes for the angiogenic ratio compared to the concentrations of VEGF and thrombospondin 1. So first, just note that thrombospondin 1 is not changing very much. Its fold change is just around 1, so it's mainly robust to these treatment strategies. But what's changing and what we're really interested in this is this wide variability in the fold change of the angiogenic ratio in the tumor specifically, as well as the fold change in the VEGF concentration also in the tumor. And so perhaps these outliers here could represent patient tumor profiles that are more sensitive to the treatment protocol. And we can go in and look at those profiles and see, well, what are the receptors that are being expressed? What is the level of expression, as to better understand why in the clinical trial there was this really heterogeneous response. So in all of these cases, the fold change is less than 1, meaning that the drug is reducing these quantities, but perhaps there is a threshold value that has to be reached. And so we can better understand that, again, using the quantitative and mechanistic detail of our mathematical model. So we can further illustrate this heterogeneous response and the distribution in the changes that are elicited by the drug treatment strategy by looking at the fold change or the response to therapy across the entire population. So I failed to mention that we ran this simulation for 1,000 times, again sampling from that Monte Carlo analysis. And so we can then run the model for all 1,000 patients which have different numbers of receptors on their tumor cells and diseased endothelial cells, and see, well, how do they respond to the mimetic alone, so that's shown in red. How do they respond to bevacizumab alone, the anti-VEGF, and that's shown in green. And then how do they respond to the combination treatment, which is shown in blue. Again, you can clearly see some outliers here. Even here, there are some outliers for the ABT510, the thrombospondin 1 mimetic. So we can look at the differences in the receptor profiles. And just to further drive home this point, we can also look at the variation in the area under the curve for the angiogenic ratio. So really the takeaway message here is that there is wide variability in the response to treatment, and what we want to try to understand is how do the expression levels of the angiogenic factors correspond to that variability in the response. And so out of all of the receptors that we changed, both on diseased endothelial cells and on tumor cells, 2 particular expression levels stuck out as being related to the response. And so here I'm plotting the fold change in the angiogenic ratio as a function of the normalized VEGF receptor 1 expression. So this is one receptor that VEGF can bind to, and also as a function of the normalized CD36 receptor expression, and that's a receptor that thrombosponin 1 binds to. Okay, so they have alternating effects, but if you increase VEGF-R1 expression, then you have less effect of the drug. If you increase CD36 expression, then you have a better effect of the drug. And still you can see there's wide variability, but overall the trend is moving in that direction. So we can really start to appreciate some of the differences and the heterogeneous response and sort of back calculate and correspond that to changes in the receptor expression level. So where are we going with this work? Well, we're really excited to not only have this mechanistic detailed model of the signaling pathways and the interactions between these angiogenic factors, but what we're really excited about is trying to link that mechanistic model to cellular level response in terms of how much proliferation there is, how much apoptosis there is, and also how much vascular sprouts are being formed when we modulate or alter that angiogenic balance. And this is work that we're starting to do in collaboration with Shannon Mommenthaler, who's here, and also Min Yu, and they're both in the School of Medicine, the Keck School of Medicine at USC. And my lab was recently just awarded an American Cancer Society grant to pursue this research so that we can combine the mechanistic model with the cellular level response. And along with Min, we can try to see how is this recapitulating the in vivo setting. So we can do a small scale in vivo preclinical mouse study to try to test and validate some of the model predictions. And we're really excited about where this work is going. So let me just summarize this first part of the talk. So I presented to you a model that helps us not only predict the concentrations of these angiogenic factors over time, but also their distribution in the tumor tissue. And where they're bound tells us a lot about the angiogenic state and how the tumor is really poised and primed in this pro-angiogenic state. And then we can use the model as a framework to study the effects of mimetics that inhibit angiogenesis, and in this particular case I showed CD47 binding mimetic that's able to strongly shift the balance to oppose angiogenesis. And then we expanded the model to look at the whole patient and have a better understanding about how the response to anti-angiogenic treatment, which is a dual inhibition strategy, how that response depends on properties of the tumor microenvironment, specifically the receptor profile. So I think we're excited about some of these applications to provide this mechanistic and quantitative insight that can perhaps help us better understand clinical observations, but also to have this tool that we can use to vary many different parameters associated with tumor heterogeneity, and even parameters associated with the normal tissue too, because we know that varies across patient populations. And so we can use this as a tool to perform in silico clinical trials and look at a wide variation in terms of patient populations and their responses to anti-angiogenic drugs. So I'd like to just move to the second part of the talk and present to you some recent work that we've done in building this preclinical model of angiogenesis-mediated tumor growth. And some of the same themes will come up in terms of the heterogeneous response and how that is influenced by not only the receptor expression level, but also in this case, tumor growth kinetics. Okay. So the work that I just showed, as well as a large body of literature in terms of experimental and clinical studies, really emphasize this point that the response to anti-angiogenic treatment is quite variable and heterogeneous. And so what we would like to have is some predictive biomarker that will help us identify before any drug is administered which tumors will respond most significantly to that treatment protocol. And so there's some recent advantages and advances in imaging analysis over the past 3 or 4 years, where now it's possible to take serial 3D images of a patient's tumor. And some recent work in analyzing those images have shown that how the tumor is growing, the kinetics that characterize tumor growth, can be used as a readout as to how effective a particular therapy has been. And so we got sort of excited about this and said, well, can we take this one step further and say, well, not just a prognostic biomarker, but actually a predictive biomarker. And so we turned to a mouse model, an in silico mouse model, mathematical model, to better understand whether the tumor growth kinetics, so the parameters that characterize tumor growth, could possibly be used as a predictive biomarker. Okay, so as a postdoc fellow at Johns Hopkins, I produced this model which represents the entire mouse. So it has similar components, this 3-compartment model, but when I was a postdoc, the tumor volume just increased exponentially. So it was not connected at all to angiogenesis or the formation of these pro-angiogenic complexes. And that really was a limitation of the model. And so what we want to do now and have done is to make the volume of the tumor a function of receptor-bound VEGF. So we have this dynamic tumor compartment, and now the volume of the tumor depends explicitly on the angiogenic signal, which we've defined as the concentration of receptor-bound VEGF on the endothelial cells within the tumor. Okay, so in addition to the differential equations that describe how the molecular species concentrations evolve over time, we have now another differential equation that describes how the volume of the tumor evolves over time. And so the functional form of this is taken from some very nice papers which describe how the volume of the tumor evolves, but we also added now this dependency on the angiogenic signal. And so we have different kinetic parameters that characterize the tumor growth, including K0 and K1. So these are parameters that correspond to the rate of exponential and linear growth of the tumor, as well as psi, which is characterizing the transition between exponential and linear growth. And then lastly, Ang0, which is the basal angiogenic signal. Okay, so we need to be able to estimate the values of these parameters. And so what we've done is to extract data from the published literature where we can fit the values of those parameters, so do a model parameterization, and fit the model using experimental data for the control growth profiles of breast tumor xenografts, specifically in mice. And so we can train the model using those measurements and then validate the model with matched measurements, where in this case now an anti-VEGF agent has been administered to the mice, and we can see how well does the model predictions match this validation data. So let me show you just one example of a data set that we extracted from the literature. So this is plotting the volume of the tumor as a function of time, and the blue information is the experimental data in the triangles that we extracted from the literature and used for training the model. And so we ran the model optimization just to match those experimental measurements about 20 or 30 different times. And so what I'm showing you here is the solid line is the mean of those best fits, and then the shading indicates the 95% confidence intervals. So we used the blue information to train the model, and then all we did next was to turn on the anti-VEGF treatment. So no additional fitting of the model parameters or tuning of the model parameters. And so the purple information is now the predictions of the model shown in the solid line compared to the experimental data in the squares. And again, the shading indicates the 95% confidence intervals. So I just want to re-emphasize that it's not the purple data that we're using in training at all. This is independent, and we're validating the model with those experiments. So the model does a good job of course, matching the training data, but also doing a pretty good job of matching also the validation data. And so this is just one data set, but we were able to extract 5 more data sets, and overall we do a pretty good job. There are these cases here, they all come from one group, and it's not a very good fit, particularly for these last two. These seem to be following a sigmoidal trend here, which the model is just not capturing very well. But what we wanted to then see is, okay, what if we had just a limited number of data sets? What if we only had measurements of the tumor growth profile without any treatment before the drug is given? So those are the blue points shown here, and there were only 2 out of those 6 data sets for which we had more than just 2 points before any drug was given. So we tried to see, can we train the model using the blue data again, and then validate the model using the purple information. And we can do a pretty good job. The model does a pretty good job of matching those validation data. Of course, there is wider uncertainty, so the 95% confidence intervals are wider, but this again is because we're using a smaller subset of data to train the model. Okay. So, but what we essentially want to understand is, how do these kinetic parameters that characterize the tumor growth, how do they correspond to the response to treatment? Can we predict the response to treatment by knowing how the tumor is growing? And so rather than just limit ourselves to these 2 data sets, we just moved forward with the 6 data sets so that we can try to make some meaningful comparisons in the estimated parameter values. So I'll show you the estimated parameter values now. So what I'm showing just first is the value K0, which is characterizes the rate of exponential growth. And for this, these are all the fits that we did for each of the 6 data sets. And I can show the rest of the data here for K1, for psi, for angiogenic, and then this ratio, which we don't fit the ratio, we individually fit K0 and K1, but just combining them together to show this particular fraction here. And so the median value is what's shown in the bar. And even from this, I'll show you a very clear statistical analysis of every pairwise combination. But even from here, you can start to appreciate that there are some differences in the estimated parameter values. For example, K0, there are some that are lower, or sorry, K1, some that are lower compared to others. Certainly for psi, some that are lower here compared to higher values, and so on. Interestingly, there's no difference in the angiogenic basal signal, so all of them vary pretty much the same across the 6 data sets used for fitting. And then just one more slide before I talk to you about this statistical analysis of those parameter values. So what we did is to say, okay, now we have these 6 data sets. We fit each of the data sets about 20 or 30 times. We can run the model with all of those estimated parameter values and then turn on the treatment when the model predicted volume reaches a particular size, and then run the model for 6 weeks with twice weekly injections, so that we can compare apples to apples in terms of what is the response to treatment. And so we characterize that response to an anti-VEGF agent, bevacizumab, by calculating the relative tumor volume, so just taking the volume at the end of the 6 weeks of treatment compared to the same time point where no drug has been given. And of course, if this value is less than 1, that means that the treatment has been able to reduce the tumor volume. And so we can do that for 2 particular concentrations of the drug, 2 milligrams per kilogram and 10 milligrams per kilogram. So first, just comparing the effects of the different concentrations. With the higher dose, then we see lower RTV values, meaning that it's able to more substantially reduce the tumor volume. But what I want you to take away from here is, even within a single dose, some of the tumors respond more significantly than others. Again, pointing out there's this variable response, this theme that's coming up several times during the talk. So then we can collect all of those parameter values, collect all of the predicted relative tumor volumes, and then do all of the pairwise combinations to compare how different are the parameter estimates from one another. So let me just walk you through this heat map. So this is trying to help you understand what are the are there statistical differences between, for example, the K1 values estimated when we fit the data to Rowland compared to the K1 values when we fit the data, or fit the model to the data from Volk in 2008. So the color of the box indicates the log to the base 10 of the p-value, and then also I indicate the significance level using different number of asterisks. So these more blue boxes and 3 asterisks mean that they're a statistically significant difference to a high level for these different estimated parameter values. We can do the same for K0, for psi, for angiogenic. All of the boxes here are purple. Remember, I mentioned that there's no statistical differences in those estimated angiogenic values, and so on. And what I want you to take away from here is that for any comparison where there is a difference in the relative tumor volume, which is the response to treatment, there's also a difference either in K1, in psi, or in the ratio of K0 to K1. So it's these differences in the tumor growth kinetic parameters that correspond to differences in the response to treatment. we wanted just to probe this a little bit more, so we did another mathematical analysis, and some of you, I'm sure, are familiar with this, called partial least squares regression, or PLSR. So it's a way to connect inputs to outputs. In our case, the inputs are the kinetic parameters that characterize the tumor growth profile, and the output is the response to treatment. We want to make this connection between the kinetic parameters and the response to anti-angiogenic treatment. And so we use this PLSR, or regression analysis, which essentially recasts all of this data onto new dimensions called principal components that are linear combinations of the inputs. And so we first want to make sure that our regression model can make sure that the predicted values for the RTV at different concentrations of the drug match the observed values. So if they do, then they'll lie exactly on the 45-degree angle line. And so we see a pretty good match here. But then also we can characterize this Q²Y value, which is quantifying the leave one out cross-validation. So how predictive is the regression model in matching data that's not used to train the model. And so we can, this can have a maximum value of 1, and so we get pretty close to 1, and so we have some confidence in this regression model. But what we want to say is, well, how important are those kinetic parameters in predicting the response to treatment? And so just one last data slide here is another piece of information that we extract from the PLSR analysis, which is called the VIP score, so the variable importance of projection score. So essentially telling us how important are these individual parameter values to the contribution to the response to treatment. And so if the VIP score is greater than 1, then that means that that variable is statistically important to the output. And so in this case, we find that it's the psi value, which is the transition between exponential growth and linear growth, that is very important contributor to the response to treatment. And so perhaps this is a way to predict the response to treatment. If we have sufficient number of data points before any drug is given, we could use the model to fit that data, estimate the psi value, and then be able to stratify the tumors according to their psi value. Okay? So here we can use the kinetic parameters that characterize tumor growth as a way to predict the response to treatment. So if I just summarize the second part of the talk, then we've expanded an existing model to have now a dynamic tumor compartment that explicitly depends on the number of pro-angiogenic signaling complexes. And then we can use the model to better understand how the parameters that define and characterize tumor growth are related to the response to treatment, and perhaps identify a predictive biomarker. And so how we're moving forward with this particular part of the project is to do a preclinical trial using our model. So have 50 different mice, each with different values for psi, and see, are we able to find a threshold above which there is a response or below which there's no response. And then also we can go into the literature and use in vivo imaging data to further test and validate the model predictions. Okay. So with that, I'll just end my talk and say that we're really trying to focus on having this predictive framework that gives us more quantitative insight into the effects of anti-angiogenic treatment strategies. So I certainly want to acknowledge the members of my lab at USC, the undergraduate students, and the graduate students who contributed to the work I presented today are highlighted in bold, as well as all the members of the lab, and certainly our collaborators, this new interaction that we have with Shannon's lab and Min's lab and other researchers at USC, and even at UCLA, the crosstown rival. And then certainly our funding sources, so we've been very fortunate to get this NSF Career Award, the American Cancer Society Award, and then also more private funding foundations as well. And I want to just acknowledge publicly, certainly my advisor, Sasha Poppel at Hopkins, who gave me this opportunity to start working in this area. Certainly in recognition of Leah Edelstein-Cachette, this is an amazing opportunity, amazing honor, in recognition of all of the work that she's done, and I hope to follow in her footsteps, and thank all of you for your attention and for SMB for giving me this opportunity to present my work. And so I'd be happy to take any questions. how do we predict that they interact with it? Yeah, so this mimetic only binds to the CD47 receptor. So you can basically take particular sequences of the endogenous protein and then put together a peptide mimetic so that it only has the binding domain for a particular binding partner of interest. And so for the CD47 binding mimetic, it only binds to CD47. And also I should note that those peptides are protected from cleavage by plasmin, so they can't be converted to that inactive form. And so in the literature, some of these peptides have been published, and we know what the association and dissociation rates are. And so we can simulate that in the model. Other questions? Yep.
So I'm not a cancer biologist. I am sort of a dynamicist, so I don't know if this is a good question, but there's something that caught my eye. First of all, excellent talk, which I should say first because I'm picking on something I don't understand. You had a calibration slide where you matched everything like really very well by tuning parameters. And there was one parameter which was off by more than a factor of 2, the proportion of MMP9 that's active. And I was wondering if there's a biological interpretation, if there's something that can be learned from the fact that you're not able to match that one thing.
Right, so I definitely left out a lot of the details. So the endothelial cells can secrete the inactive form of MMP9, so in this pro form, and then MMP3 can actually activate MMP9 so that it's able to cleave VEGF. And so there is possibly something that we're missing in terms of that, that particular sequence of events, and we haven't provided all of the mechanistic detail or put in all of the mechanistic detail in the model about how pro-MMP9 becomes active. So it's perhaps something that we're missing some of the detail there that could then help us better match the percent of MMP9 that is active, and that has been measured experimentally. But yes, thank you for catching that, and it's certainly on our radar too.
Yeah, that was a lovely talk, Stacey. So a lot of people that think about this, from my understanding, is there's a spatial aspect to the tumor dynamics. So you want to maybe comment on maybe that loss of generality potentially by not having that, and that could maybe account for things like the MMP and other aspects.
Yeah, so you're right. We've thought about like having maybe some, so not doing the full PDE simulation, but like having some, within this particular compartment, compartments to say, well, have some localization of where the molecules are interacting. But I feel that it's something that we're missing right now. It's just really hard to implement because of the time scale over which and the complexity of the number of species that we're including right now. So we're looking sort of at longer time scales over weeks and days. But certainly that is a limitation that we can start to look at.
We have time for one more question.
I'm afraid it is a little bit perhaps related that in your compartmental model where you have blood and the tumor, you have, I assume, transport parameters which would tell you the transport of VEGF, let's say, from the through the blood. So do you think that those parameters could be characterized by the morphology of the vascular network? So like from imaging you could determine the, let's say, whether how what is the vascular structure and perhaps tune your your transport parameters accordingly.
Right. So for the whole body model, we have transendothelial permeability, so across the endothelial cell surface, and then we also have lymphatic flow from the interstitial space of the tumor to the blood. There is also lymphatic flow from the tumor, but those lymphatic vessels are very compressible, and so they're actually not functional in most tumors. But, right, so the transendothelial permeability, we can take the values of that from experimental data. And one thing I think also that we can take even a step further is that the permeability changes as the anti-VEGF treatment is going along, which we don't include here. So we have a single value for the permeability, and it's not varying and improving with treatment. So certainly it's something that, again, we can talk about. Yes, yes. So the tortuosity of the tumors, the leakiness of the vessels, like we're trying, I mean, there's so much that we can incorporate, but yeah, this is really, I feel like a good start where we can expand even more.
Great. Well, thank you very much for a wonderful talk, Stacey.
Thank you.